answers to these questions yet, because all the populations differ and all these different variables. And so a lot more work needs to be done to untangle these different factors. So I'm just going to tell you a little bit about how the microbiome relates to genetics and health. And you might be wondering whether all this diversity matters. And for mice, it certainly does. So with Jeff Gordon's group, we've been studying these leptin mutant mice, which are much fatter than a normal mouse. They also have a radically altered microbial community in the gut relative to wild-type mice. And so you might be wondering whether that change in the bacteria is a cause or an effect of the obesity. It turns out that you can take the community from the obese mouse, transplant it into a germ-free mouse that's genetically normal but has no microbes of its own. That other mouse will also get fat. And the same is true for a different model, the toll-like 5 receptor knockout mouse, which is lacking a normal component of the innate immune system that we worked with Andrew Gewirtz on. So you can see that it's a lot fatter. It's got larger fat pads in males and females, higher triglycerides and cholesterol in the blood, high blood pressure, and again, a radically altered microbial community relative to a wild-type mouse. But in this case, what's going on is it's not an energy balance phenotype, so the OB community is more efficient at extracting nutrition from the diet. With these mice, what's going on is something different. It turns out that they actually eat more than a wild-type mouse does, and the same is true for a genetically normal mouse that you transfer those microbes into. So in this case, it's having a behavioral effect rather than a metabolic effect. And John is going to tell you a whole lot about additional behavioral effects of the microbiome in the last talk. But what this means is remember that you have a whole lot more bacterial cells in you than you have your own cells, and they may potentially be outvoting you when it comes time to make choices at the cafeteria. So you might be wondering if this mouse stuff extends to humans. Jeff Gordon's group has shown that the longer people are on the longer obese people are on a diet, the more they come to resemble lean people microbially, and then the more weight they lose, the bigger the change in their bacterial community. And Dan Knights, who was a really talented grad student in my lab, who's now working with Ram Xavier at Harvard, was able to show that we can classify people as lean or obese with 90% accuracy based on their gut microbes. And the reason why this is really impressive is that with genome-wide association studies, there's a whole lot of human genes associated with obesity, but you can only predict whether someone's lean or obese with about 58% accuracy based on their human genes, whereas you can do it with 90% accuracy based on their bacterial genes. So can we actually exploit this to manipulate the microbiome to cure disease? Well, one really interesting question is whether we can do something not just about obesity, but about malnutrition. So in another project with Jeff Gordon's lab, funded by the Gates Foundation, we've been looking at populations in Malawi and Bangladesh. So this photo was taken by Tanya Yatsenenko, one of Jeff's grad students at the field site we work with in Malawi. And although food is scarce in this population, you'll notice that they do, for example, have access to cell phones. And the remarkable fact is that there are now over 5 billion activated cell phones on Earth, which is really impressive when you consider there's only about 7 billion people. And a lot of the reason for this is Moore's law. The decrease in the cost of computation means that processing the digital signals is effectively so cheap that it's free. And this isn't just true in Bangladesh, in Malawi, for example. So I took this photo in Bangladesh last year in Dhaka, where even in relatively poor parts of the city, you can buy a sizable hunk of a cow, and then right next to it, you can get your cell phone minutes recharged. And so, what we're looking at here is the decline in the cost of computation, which is this white line. But in the time that computation has dropped in cost by a factor of about 100, DNA sequencing has dropped in cost by a factor of about 1 million. So the question is, can we come up with a kind of personalized medicine based not on subtle differences in the human genome, but based on these very obvious differences in the human microbiome? And so we're starting to do this now by taking microbes from malnourished children on the Gates project, and then from obese adults on an NIH project, transplanting them into mice, and then looking at how those mice gain or lose weight, depending on whose microbes they got. And the results from this are just beautiful. So this axis is time, this axis is the microbial community. And so 5 mice colonized from the same human subject start off looking pretty much the same on mouse chow. When you switch into the Malawi diet, which is about 90% corn and 10% leaves, you see the sudden change in the microbial community that you can just model with an exponential decay curve. When you give them the ready-to-use therapeutic food, so the peanut butter-based supplement that's used in the clinic, they start to recover. but after 2 weeks, when you switch back to the Malawi diet, they go back to that same state. And with Jeff's lab, we were recently able to show that mice with individual transplants, either from healthy children or from children with kwashiorkor, a severe nutritional deficiency, respond very differently. So the mice that get the kwash community lose weight very rapidly and they're really sick. But then when you supplement them with a therapeutic food, you get this rapid improvement, whereas the mice that are colonized from the healthy child show very little change. And then this is coupled to a very profound change in the microbiota. And so what we can see is that the responses in mice can potentially tell us something about what will or won't work for an individual. And so obviously the germ-free mice, where you're raising them in a bubble and keeping them away from everything except the microbes you supply, are very expensive. But the idea is to move from this to an in vitro system and then ultimately to a computational system where you can predict for the microbes what diet or drug or other intervention will work for individual people. So basically the idea is to develop a restoration ecology of the gut. And it's kind of like your lawn, where the idea is that you might have an initially flourishing ecosystem that's then ravaged by diarrhea or antibiotics or other issues. And then left to itself, maybe you have weeds running wild if you just let whatever recolonizes recolonize. Then the question is, what should you do? Should you seed in good microbes with probiotics? Should you promote good microbe growth with prebiotics? Or should you use bacteriotherapy, like has been very successful for Clostridium difficile infection, where you're transplanting the whole ecosystem? And then to what extent can you restore something approaching, but maybe not quite reaching, the diversity of the original ecosystem? So what have we learned from all this? Well, we've learned that different body sites, different people and different populations are incredibly diverse in terms of their microbes. We've learned that the microbes can cause, as well as being the effect of, really large differences in phenotype. And we've also found that the prospects for personalized medicine based on the microbiome are looking really good, just because there's so much diversity there in their microbial genes, as opposed to your human genes. And you might be wondering how you can get involved with this. And so Time Magazine, at the start of 2007, made you, as in the producer of user-generated content on Wikipedia or YouTube or other websites, the person of the year. And it's literally true that you can do this for your microbes now. So for example, if you go to Google and you type in American gut, and I'll just do that, what you get as your first link is the link to this project where you can actually start to make use of this information, of these techniques developed in the Human Microbiome Project, and find out about what's in your own gut microbiota. And so if you click there, this is a crowdfunded campaign that you can potentially contribute to, either in terms of money or in terms of microbes, or both, to really help to understand what these differences in the microbial community are doing and how they correlate to diet, physiological state, and a whole lot of other things. So, so what's really exciting about this is it gives you the opportunity for the first time to participate in this microbiome research, as opposed to just reading about it in the newspapers. So what can you do? So basically, there's a lot of these questions about the effects of diet, lifestyle, pets, kids, and so forth that are largely unknown. And by signing up, your microbes, or perhaps those of your child or pet, might help us understand a range of diseases that are now being linked to the microbiome. So with that, I'd just like, and we will of course protect your privacy to the extent possible, and at least so far, we don't think there are any risks associated with making your microbial genetic data available. That's very important to mention, and all of this work is approved by our Institutional Review Board. So with that, I'd just like to thank the large number of people in my lab, both current and former members, and our huge number of collaborators across these different projects. And I won't read all these names, but I just want to point out how highly collaborative microbiome research is. And I think that's a theme that will emerge in the other talks. So with that, I'll hand things over to Eric and just say thank you for your attention.